So the next couple of speakers are going to be clinicians and researchers who are doing exactly what Dr. Bilski mentioned at the end of his talk, trying to figure out what's actually making these tumors grow, appear, and how do we cure them without having to do some of these heroic things that he was talking about earlier. So Dr. Hameed is a pathologist. She is a person, you heard about the importance of actually making the diagnosis and how difficult that can be. She's the person who does that and does that extremely well. And she's also spent a lot of time studying these tumors, trying to figure out what genetic changes are making them grow and appear. So she will talk about that.
Thank you, Ilya. I want to thank the Chordoma Foundation for arranging this conference. I'm a pathologist, and I don't see patients, and I don't see patients' families. And often, when we look at the disease under the microscope, we are always taught that don't look at it as a slide, it's a patient there. And we teach our residents the same, and if we don't have the opportunity to see you, I consider this as an honor and privilege to be able to talk to you about this disease. So what I thought I would do is actually, I'm going to start from the beginning. Everything starts in an embryo. So this is a diagram of a chick embryo. And you can see the notochord is a structure, which is a rod-shaped structure, which sits between the neural tube and the gut. And it starts at about 3 weeks of embryonic life from the mesodermal cells, and it extends itself from the head to tail. So it's along the entire axis of the embryo. And notochord is a very important structure. It secretes a protein called sonic hedgehog homolog, which is very important for organogenesis, formation of the neural tube and motor neurons. But once these neural tube gets formed, the notochordal cells have no role. They are fated to die during the development. But as a pathologist, we always want to see how everything looks under the microscope. So if you look at the notochord under the microscope, these are the notochordal cells. They look like fat cells. They have these large vacuoles within their cytoplasm, pretty much empty spaces, and they have small nuclei. And these things obviously disappear. We don't need them anymore after the neural tube gets formed. And they get replaced in mammals by vertebra and vertebral discs. So in the early life, in the intervertebral disc, small remnants of the notochords remain, what we call nucleus pulposus. And about 3-4 years of age, these die out as well. So there shouldn't be any notochordal cells in our body. But it so happens that some of us retain these remnants within the vertebral bodies and within the intervertebral disc. And it can be seen about nearly 20% of, if you do autopsy series and look at the vertebrae. And that was actually shown in this study by Ilya and Dr. Bilski and us. Over a period of 8 years, they collected about 8 patients who have these notochordal, what we call notochordal remnants. So here is a C6 vertebra, and this is MR. You can see that there is a dark intensity signal here and a light a bright signal here, which is a presence of the notochordal remnant. It's also in the sacrum. Sometimes these patients can have multiple notochordal remnants within their spine. And they are, in a CT scan, they are very subtle. Usually you see them as sclerotic changes. They tend to be inside the vertebral body. They don't break out of the vertebral body. And these patients were all biopsied. So we confirmed as pathologists that these are notochordal remnants. And they have very similar histology as you saw on the embryo. So they have these large cells which have this vacuolated or empty cytoplasm and these smaller nuclei. Chordoma, on the other hand, is derived from the notochord. We consider this as a slowly growing malignant neoplasm, but as Dr. Bilski talked about, as slowly growing this can be relentlessly recur in a patient after surgery. So treatment, as we already heard enough about the treatment, I don't have to say much more. There is a problem of local failure. So many times just surgery alone does not cure the patient. And from a biological perspective, there are some associations with chordoma. Certain inherited disorders, this is tuberous sclerosis. It's a disease caused by mutation in 2 genes, TSC1 and 2, and these patients have multiple benign tumors in the skin, in the brain, in kidney, and some of these patients actually develop chordoma, and most of them are in children. And we also know there are familial chordomas, which are very well described now. And we have seen the radiology of, of chordoma itself. Unlike the notochordal remnant or benign notochordal cell tumors, chordomas tend to really break out of the bone into the soft tissue, which is what makes it more difficult to take it out en bloc. And histologically, again, it is a spectrum. So when you look at this from this power, you see, yeah, you do see those cells with the empty cytoplasm or vacuolated cytoplasm, but they are a little bit different. What you see here are this lobulated growth pattern. There is a matrix to these cells. When you look at it closely, this blue matrix is made up of mucin and glycogen, and that is something they seem to acquire as a malignant tumor. And the cells themselves then start looking larger. They do have this vacuolated cytoplasm. These are called physelliferous cells. Physelliferous in Greek means bubble. So, and that's characteristic from a pathological point of view, how to diagnose this disease. And we use markers to confirm sometimes this is a chordoma versus some other tumor. And for a long time, the genetics of this was not well understood, except that this is genomically pretty unstable tumor, until this paper was published by Dr. Adrienne Flanagan. You all probably already know her very well. She's contributed tremendously to this disease by doing expression analysis with chordomas and chondrosarcomas. It was found that brachyury, which is really a crucial regulator of notochordal development, is consistently expressed in chordomas. And we use that as a diagnostic marker when we have to diagnose a patient's disease as chordoma. So you can see that this brown dark staining is in the nucleus of this chordoma, which is by brachyury. So the role of brachyury in chordoma has been still under a lot of research, but it has been shown that in familial chordomas, you get gene duplication of brachyury. Some sporadic chordomas have amplification or extra copies of brachyury, but brachyury has not shown any mutations in chordoma. As Dr. Bilski already pointed out, there is an association with a single nucleotide polymorphism. That is, you inherit this from your parents, and people with chordoma tend to have this particular SNP in their brachyury gene more commonly than those without. And it does play a major role in the disease, is shown by the fact that if you take chordoma cell lines and you silence the brachyury, they stop growing. So obviously, this has a very important role, is an activator of transcription in these tumors. And the difficulty really is it's a complex transcription factor, and it's very difficult to target as a single molecule, a single gene. So we, about 2 years ago, we started this chordoma study. First, we wanted to look and see what kind of samples that we have in our tumor bank. So we found 17 frozen tumor tissue of chordoma with matched normal from same patients. And we also have 6 without matched normal. Here is just the clinical summary of these samples. Most of them were male patients. Most of the tumors in the sacrum, Dr. Boland does majority of our sacral chordomas. We had 4 patients who had recurrences of their tumor. And as we heard earlier, some patients did undergo radiation therapy, pre, post-op, or combinations. And we did have a follow-up up to 17.5 years and with a median of 3 years. In the pelvic recurrence, so when we give radiation to these tumors, and if we want to take them and culture them and grow a cell line, radiation already has had an impact on the tumor, so it's very difficult actually to grow. But it so happened one of our pelvic recurrent patient did not have radiation in the past, so we were able to harvest the tumor for culture and cell line development. And when we do any study on any of these tumors, first we want to make sure that there is adequate tumor within the tumor tissue, because tumors themselves undergo spontaneous necrosis. So when you undergo necrosis, you cannot really study them towards any type of genetic or other changes. So as a pathologist, I would look at these tumors to make sure there is viability in these tumors. So we made sure that at least 70% of the tumor was viable. And we decided to do an analysis where we do like a genomic profiling just to first understand what is the whole genomic landscape of this tumor. So we took two technologies. One is the SNP array analysis, which look at the whole genome profile of changes in the chromosomes, which are really broad changes. And then we also wanted to do our next generation sequencing platform, which looks at cancer genes, which are, some of them can be targetable with drugs. This is a test which is clinically used in our lab. So this OncoScan SNP array is a very robust technology. You can look at genome-wide copy number analysis of the entire tumor. You can look for genes which are amplified or an increase in their copy number, or genes which are deleted, that is lost. And then you can also look at what's called copy-neutral loss of heterozygosity, which means you lose a portion of your chromosome or allele, and then the other one duplicates itself. So it looks like you have a normal chromosome number, but really it is an abnormal chromosomal number, which is what we can detect by this technology. And then you can detect some very small number of mutations. So this is our summary of our copy number changes by this SNP array analysis. And even when you look at it without knowing anything, you see like a pattern, right? You can see the red is loss of a gene or loss of a chromosome arm, and the blue is gain. So these tumors form a pattern. They have recurrent losses and gains. And even as when you see here, you see there's more red than blue. So they seem to have more chromosomal losses than gains. And this is just an example of one of the samples. And you can see that this central line would be a diploid or a normal chromosome. Chromosomes are numbered from 1 to 22. And then when you have something below 1, which will be a loss, which is also reflected in this track, which is a allele track, which shows that there is loss of the middle track, means you have actually imbalance in the allele. And then gain is shown by something above 1. So we can actually look at this and say, okay, how many are lost, how many are gained? And we also saw other changes. This is a chromothripsis is now a well-known phenomena. This is an interesting type of chromosomal abnormality where in some tumors, some of the chromosomes will completely shatter themselves and then put themselves back together and function in the particular cell. And that happens in chordomas, sometimes it happens in osteosarcomas, and we did see that in about 12% of cases, and the way we identify that is there is a loss and gain are multiple sort of breaks within the chromosome, which tells us that this is a chromothripsis that's happened in this particular chromosome and this particular tumor sample. So what are brachyury? So when we looked at our samples, we found one sample which showed actually brachyury being amplified. That means there's a lot of gain in its in the chromosomal arm of that particular region. And we only saw one sample which had that. So our summary of this SNP array analysis tells us that this is really a tumor where there is multiple chromosomal losses. Chromosomal gain is only seen mostly in chromosome 7. And we do see this chromothripsis. This is shattering and bringing back putting back the chromosome piece together. And certain genes are much more focused or lost, just like the CDKN2A. This is a tumor suppressor gene, very, very important for cell cycle, is lost in many different tumors, and chordomas seem to show a homozygous deletion, that is both copies are lost in about 22% of these tumors. So when you look closely into the changes that we see, now we look at individual areas within the losses and see what can we find, what kind of genes are there that may give us some clue as to where to take this next. So as we saw, about 65% had loss of 3P, the minimal deleted region in this area overlapped certain genes. These genes are SETD2, PBRM1, and BAP1 gene, and these are, as Dr. Bilski pointed out, these are involved in chromatin remodeling. They are related to structure of the histone complex in us in general. So that seems to play a role. And then we have loss of 9P, which is really this CDKN2A gene, which is very much involved in cell cycle. It is a controller of cell cycle. It makes sure that the cells doesn't simply divide beyond what they should do, and when we lose it, obviously there is more proliferation. And then we have chromothripsis, which is a much more difficult phenomena to study because you have a chromosome which is shattered and brought back together, and so there are multiple different genetic rearrangements and aberrations that happen. And next, what we wanted to do was, is there a large mutational profile in chordomas? So this particular test that we do, we take the tumor and the normal, we break them in the DNA into multiple pieces, and then we label them with barcodes for individual DNA, and we actually hybridize it with a whole bunch of cancer gene probes, and then we sequence in a very highly efficient machine. So we can analyze these sequences to a reference genome, which is a normal human genome, and then we compare it to the patient's normal DNA to see what is lost or what is gained in this particular tumor. So you can look for single nucleotide changes, which are what we call base substitutions. We can look for insertion and deletion. That is, some sequences are inserted into a particular part of the gene, or some are deleted, and then we can look for big rearrangements in these. So these are all done in one test, as that's because we can sequence this in a very efficient manner. So just to show you an example, here is a tumor and now normal from the same patient. So of course, all our genomes should look alike. So you can see that they all line up and look alike. But if there is a difference between the tumor and normal, you can just pick it out, because this tumor has a mutation in this EGFR gene, and it is not present in the normal. So that means it's acquired in the tumor, so we can use it if this is something we can target. If there's something which occurs in 10 of the chordomas that I'm studying, that is very relevant, that we can do something about it. So the list of genes is 341. Almost all of them are cancer-associated genes. Some of them are now targeted drugs are available. So what is our outcome of this study? So it seems that in chordoma, these are the genes that we found to be mutated, but the mutation number is quite low. So at the most, there are like 2 mutations in a single gene. Majority are single mutations distributed along the different samples. So we had 14 samples which showed mutation, but they are not a large number. So when we combined the copy number changes, the long chromosomal changes, and the mutation analysis, we see a pattern. So what we see here is, this is, as I told earlier about SETD2, this is a DNA methyl transferase. It's an enzyme which causes methylation within the histone of the cell. Histones are proteins which actually pack the chromosomes together, because our chromosomes are really very long. They have to be packed together within the DNA, within the nucleus, and this is where the SETD2 plays a big role. So whenever its role is to cause methylation in a particular residue, which leads to opening up the chromosome, opening up that particular chromatin, so more genes can come and bind to it. So if there is something wrong with it, if it is deleted, if it is mutated, perhaps that whole mechanism is knocked off. So some other changes happen. And interestingly, there is another gene which is also in the chromatin remodeling complex. It is PBRM1, also having deletions. The reds are deletions in many patients, in many samples of the chordoma. And we see again the CDKN2A, which shows a homozygous deletion or hemizygous deletion. We saw the SUFU is a gene which is involved in it's a regulator gene for that sonic hedgehog pathway. So sonic hedgehog is a secreted protein at the embryonic stage of notochord. So this gene is a regulator of that pathway. It seems to be at least deleted in some of the samples. And then we saw also TSC2 deletions. Again, there's a association tuberous sclerosis complex with chordoma, and we do see some samples with TSC2. So there are some changes which seem to show something towards certain pathways which are more actively involved in the tumorigenesis of this neoplasm. It seems to be an epigenetic phenomena much more than a mutational phenomena. Actually, this particular article showed that if you take, this was all in carcinomas, but if they took 12 different carcinomas by the TCGA project of 3,000 tumors and found that tumors have two different pathways. Some are high-level mutations, some are high-level copy number changes. And chordoma seem to belong to this group where you had more losses and copy number changes, which leads to activation of pathways that we have to explore further. So our summary, as you see here, the copy number changes really stand out. And even within that, you can actually identify some loss of function in specific genes like SETD2, PBRM1, CDKN2A, SUFU, and TSC2. And these are involved in certain signaling pathways like sonic hedgehog, methylation, and also some of the tyrosine kinase pathways where TSC2 actually is involved. And recently there are articles, there's one article which has come out about SETD2's function, not generally just chromatin or putting together the chromatin, but also perhaps in DNA repair and replication within the cell. And that was shown in renal cell carcinoma because renal cell carcinomas are highly, the chromosome 3p area is highly involved in renal cell carcinoma. So by many experiments, they showed that this SETD2, which is lost in many of these tumors, actually is involved in DNA repair. So what can we do then? There are some DNA repair inhibitors. Could they be as emerging as new strategy or new targets to treat these tumors? It's something, obviously, more research has to be done, but there's a way that you could think perhaps that is an area to explore next. And not only that, there is another article which came out recently that there is cooperation between these chromatin remodeling or chromatin-associated genes, especially the SETD2 and PBRM1, which we saw. Many of those samples show both loss of both SETD2 and PBRM1, so they can co-occur in a tumor, not just by chance, by even they seem to occur together. So there is some functional cooperation between these two, and perhaps that will lead to some future discovery of where we can find targets. So our genomic profiling overall has shown us that this tumor is a high copy number alterated tumor. Yes, loss of SETD2 we are seeing in a large number, and is there a chance for a DNA repair inhibitor in the future? We have, our cell line is still live and viable. We have done our copy number SNP array analysis of the cell line and the tumor from which the cell line was derived, and it's exactly identical. So we know the cell line is keeping up its initial genomic changes. And at this point, we are trying to grow xenograft on the cell lines, and these are very slow-growing tumors. It does take time to sometimes to get these xenografts to grow. But there is opportunity to use the cell line towards perhaps further functional studies to know if there is any involvement, how perhaps a DNA repair inhibitor, would it really inhibit these neoplasms? So I'm going to kind of end here. This is basically the foundation is trying to do every possible way to come up with the whole genome sequencing, exome sequencing, and to find every possible genetic abnormality to put this puzzle together, which is what we are trying to do. And obviously, this needs not just one, many hands to put this puzzle together. And I would like to thank Dr. Boland inspired me to start working on the chordoma. And I have many people in pathology, Dr. Lu Wang, who works very closely with me, Dr. Zahir, who's a bioinformatician. Yesterday, he was going away, and I asked him, I need you to put some data together for me, and he did before he left. And obviously, all the neurosurgery Dr. Brennan, who will show his data. We are all in it together. And I just want to end with one thing. I met Josh last year when we had a Chordoma talk here, and I didn't know anything about him. It so happened at dinner, I spent like 15 or 20 minutes talking to him, and I was I can see how inspiring he is. And I tell all my friends that he is an inspiring young man. I couldn't believe when I talked to him that he's already gone through what we're all talking about. So I want to thank the foundation for having me here. Thank you.
We'll have questions after. So as Dr. Bilski said, understanding the genetics is really complicated, as you all heard. The way he explained this to me, and this will be the introduction for Dr. Brennan, is so as you heard, we all have genes. Genes code things, how tall we are, what color our eyes are. And for decades, we've been looking at these genes, trying to really interpret what is this code, and aside from being able to predict somebody's eye color, is there a clue there to who's going to develop a tumor and how it's going to grow and what's actually making this tumor grow? So for decades, people have been trying to come up with ways to really break down this code and come up with a way to sequence it. And probably 20 years ago, it would take a year and probably millions of dollars to interpret one set of one person's code. And very recently, in the last probably 5 years, it became a lot easier because computers are better and everybody's smarter. And everybody was really patting themselves on the back because it probably takes maybe a few hours and maybe a couple hundred or $1,000. The scientists will correct me in this, to do this. So it became much easier. So people are very excited about the fact that we're going to figure this out right now about every single cancer and everything else. And then things got complicated because people realize that it's not just a matter of what the code is, but how it's read. And these genes, they're coded on chromosomes, and then there are all these mechanisms that make these things read in different ways. So somebody may have the same exact code, but then we look at them and they're a different person with a different problem or no problem at all. And so Dr. Brennan is going to talk about that aspect of it. So not only how do we just break down this code, but how it gets read and what effect that has on the development of tumors.
Thank you so much for coming and for the foundation for inviting me. For those watching on YouTube, that is not I'm not so vain that I put my own picture up. But that's the neurosurgeon who really inspired our work in the last year and brought this to my lab and really brought my attention to great opportunities here. His name's Nelson Busasade, and if any of you have a chance to curbside him, he would be happy to talk further about this work. So I have no conflicts of interest to disclose, and no off-label uses of drugs will be discussed. That's a boilerplate that we put up. Actually, I wasn't asked to put it up, but I put it up because it's boilerplate and because it's kind of depressing. What it means is I'm not here to talk about any new drugs that we're developing that are going to hit clinical trials right away. And what I want to do is fill in this area that I think Ilya described as the more complicated area, an area where technology is now beginning to deliver, and that's in the epigenomics, the part of the delivery of genes out to the cell that's not necessarily related to the code. And as technologies are now progressing quickly, we're beginning to find that that domain points to really specific and different potential vulnerabilities. And I would make the argument, and I will today, that I think important effective drugs for chordoma probably are already out there, and we just don't know which ones they are yet. So raise your hand if you're comfortable explaining the difference between DNA and RNA. No, don't raise your hand. This was a question, it's in quotes because it was a question by James Treefel and Robert Hazen, who were very interested in scientific literacy. They're both expert PhDs in geology and physics. And they asked it of their colleagues, 24 PhD geologists, geophysicists, and physicists in the audience, and only 3 raised their hand and were comfortable with just explaining what is one of the landmark descriptions of biology. So I raise this to say that even the simple questions are great questions, and if you look around while I'm presenting this and think everybody else understands what I'm talking about, no. And if you do understand and feel comfortable with it, you're in a super select group along with the 3 out of 24 physicists. But I am open to any questions, please, at the question session or afterwards, just curbside me. So we talked a little bit about the way that DNA is kind of packed up into chromatin and the role of these histones, which are like the little turns, they're like little spindles around which you can turn the DNA to pack it together. And as you know, the base pairs here in the DNA run out sequences, and the sequences end up producing proteins out of amino acids. And if you get a point mutation, you get a different amino acid in the protein. Or if you get a deletion, you get a new protein where some of the amino acids are missing, or it could just wipe out the protein production altogether. There are other things to look for in the genome just based off of reading these spellings. I mean, one of them would be to look at how the gene product, the mRNA, the RNA, is put together from the DNA, and that's done by those putting together those exons that Dr. Hameed showed, and they could be done different ways, and that would be called alternate splicing. And you could read these with sequencing the RNA instead of the DNA. And you could also identify cases where there were translocations or fusions. One part of the genome gets stuck to another part, and that's a really common event in some kinds of cancers. There's chromosomal amplification, where a segment of the region of the DNA gets run out over and over again, and that just produces tons and tons of RNA, and that produces more products. So then there is gene expression, that is the mechanism by which the genes are run off into RNA and how that's regulated. And that becomes very kind of deep, complicated for reasons which I'll go into. And one of the ways that we can actually easily read with sequencing now is DNA methylation. So methyl groups get added to DNA, and that can block, or in some cases enhance, the pace at which a DNA gene is transcribed into RNA and onward. So I just want to put this up because I think it's a great picture that most people haven't seen. That's the human genome. It was printed out into books after it was after it was sequenced in 2000, 2001. It gives you a sense, 1,000 pages in each volume, 116 volumes, and that's one half of the genome. That'd be, let's say, what you inherit from one parent, and then another book, very similar book, from your other parent. But this is extremely concrete. You can download the sequence on your laptop. But making sense of it has turned out to be really difficult. So after the human genome was done, the sequencing machines were ready to go, and things got a lot cheaper, and everyone decided, let's sequence cancer now. And we want to leverage one thing. We don't want to have different groups looking at different tumors and coming back and commenting on their piece of the elephant. We want to put the same tumors through everything and get a final answer as deep as technology could go at the time. And that was the Cancer Genome Atlas. The idea was to do as much as possible on each sample. Now this we did not have and do not have yet for Chordoma. This was a government-funded project. It was a $100 million pilot project for 3 cancers. And the first cancer through was glioblastoma, and that was a cancer that I was working on. And I want to take you through a little bit of what we learned from that project, because I think we can leverage some of those dollars and indicate where maybe the most valuable pressure points are in terms of profiling chordoma. So, big project, multiple centers. The cost came in not only into the assays, but all this infrastructure to collect the samples, to have pathologists review them, to derive all the DNA, the RNA, send it out. It was and handle all the data. Very, very extensive and time-consuming. The goal was to sequence about 500 patients per cancer for 3 cancers. So this is the timeline for when the double helix was discovered. That's each box is a year, and by that many boxes, from 1953 to here, that's when the human genome was started, and it took that many years to complete the human genome. 1 year of break, the Cancer Genome Atlas starts, right? And so we sequenced 12 individuals, or not we, but the NHGRI and competitors sequenced 12 individuals in this time. In this time, they've sequenced now many thousands of cancers. Okay, so very fast pace. And this is the kind of data that comes out of these projects. They're basically pictures of tumors where the mutations are marked out here, different kinds of mutations. And you can see the big bars here are the genes that are the most mutant. And this was for glioblastoma, and it just gives you a picture of a tumor with a lot of mutations, very recurrent mutations, and it's not the picture we see in chordoma. If I take that GBM picture and I say, okay, well, what are those genes doing? What pathways do they go in? It's remarkable, actually. If a patient walks in the door with a glioblastoma, 90% of the time you can find an absolute red meat mutation in one of the genes driving a growth factor pathway or regulating it. And about 90% you can find a mutation affecting a tumor suppressor pathway, P53, and about 80% of the time one affecting RB. And not only that, they have different transcriptomal signatures and different kinds of tumors that they look identical, but they have different transcriptomal signatures, different mRNA signatures. And if you look, they actually kind of break out. So one set of patients seem to choose from one set of the menu, and another set of patients seem to choose from a different set, and et cetera, right? And we have drugs for a number of, especially the growth factor pathway mutations here in yellow. And we were set up to think we could parcelate our patients into individuals based on their mutations and give them the drugs and see results. And I will tell you that it has been incredibly frustrating and largely a failure. I would say almost entirely a failure in our clinical trials. Here's just one example. About 60% of patients have EGFR mutation, very clear. And yet, if you give them an EGFR inhibitor and compare them with historical controls, there's no difference. And it doesn't seem to matter whether the patient had the mutation or not. Now, this is depressing data. That happens to be the state of where we are at the very moment. I think it's going to change, but it's going to change by understanding the biology of the tumor better. There are other tumor types that I could show the same kind of data, and the mutations respond beautifully. So I think we can't predict which tumors are going to be sensitive to inhibiting the mutations that we find in that tumor. Okay, if that didn't work and you have to study glioblastoma, you have to go back to the book. And so that book represents the template for all of your cells, and all of your cells in different tissues are different. So there has to be a way of the skin cell knows it's a skin cell, and a neuron knows it's a neuron, and all of that is epigenetics. The same template is there, but it's just been modified so that certain parts get read out and produce a skin cell, other parts produce a neuron. And part of that epigenetics, so this is much more complicated to study, and that's why I said there's been a technological lag behind sequencing the genome. And I will confess, I am not an expert in studying this aspect of epigenetics. That's leading edge work done by specialists throughout the country. But we can start to use the assays that they've developed on tumors like chordoma to investigate. So one of the ways that it's regulated is in histones, and there are these, again, these little spools around which the DNA is wrapped. And when the DNA is wrapped around them, it's compacted, it's ready for packaging into a chromosome, and it's no longer ready to be read out into mRNA and to make gene products. So whatever opens up those histones opens up that DNA to be read, and it turns out there are complicated marks that get added to the histones to do this work. So here's a strand of DNA. You see the little spools that are binding it up, but there's an open area here where there's a gene, and then there are other areas nearby where there's guidance machinery to pull in enhancers that pull in transcription factors that then tell the gene to be transcribed more and more. And because this has to be regulated for every different tissue, think of the notochord, right? It's got a very specific and busy job very early in embryology. It has a very specific regulatory mechanism, of which brachyuria is undoubtedly one of the keys. Zooming in a little bit, I just want to point out one thing. This is a complicated slide, but I just want to point out these things. These are little tiny marks that are attached to the histone. So the proteins that make up the histone are kind of hanging out, and there are enzymes that will come and decorate them with specific signals. And those signals turn out to tell the histones whether they should unravel or not, or whether they should recruit in other proteins to start transcribing or to bind to enhancers. So what are these little signals? So they're the point of investigation, and we're finding more and more about them. Here are some examples of different amino acid residues, the 4th, the 9th, the 27th, the 34th, and the different kinds of enzymes that are in the body naturally that either attach the decorations there or take them off. Very complicated picture. But when we looked in GBM, we found a really remarkable story. And again, I say we because I'm a clinician, and I was very excited. It didn't matter whether I found it or somebody else. So IDH mutation was identified, and a mechanism emerged which suggested that it was really altering the epigenetic state of the tumor by modifying DNA methylation, but also modifying the histone marks. And then a blockbuster was the finding of mutation of the histones themselves in a subset of tumors, never seen before in any cancer, and it was found in a subset of glioblastomas. And very prominent, very prevalent. And it turned out you could take glioblastoma and you could just divide it up by the different epigenetic mutation that was discovered. So if they had a histone mutation, either K27 or G34, they were one kind. And they really were different. Actually, these are pediatric glioblastomas, only found in kids and very young adults. And the IDH mutant tumors were found in young adults, but not older adults. At least that's typical. They have a better prognosis. They're different tumors, but I will tell you that all the tumors on this list look the same under the microscope for the most part. There's a large group of adult primary GBM. That's what we studied in TCGA. We did not have a signature epigenetic hit. So we knew epigenetics is important in glioma, but we couldn't figure out and haven't figured out yet what the key hit might be in primary GBM, the most common kind of GBM. But when we go back here, there's a clinical trial treating that epigenetic alteration, more than one, in IDH gliomas. And shortly after discovery, 2 years after discovery, there are at least 2 approaches that are moving towards clinical trial for the K27. So they're seen as potential therapeutic targets because they look good preclinically. So we were stuck with the primary GBMs and having to answer the question, what is the gene, if any, that remodels chromatin that's mutated? Now, the subset that were IDH mutant, we got that. But the rest of them, we had to look at a set like this. That there were more mutations among this family of chromatin remodeling genes than chance alone. So it clearly was important. But actually, there's no way to really know which one of these is a key mutation and which is just by chance alone. You have to understand what the enzyme does, what the mutation does, and what the biology of the tumor is. I'm going to say that's where we are in chordoma. So chordoma appears to rise, as Dr. Hameed said, from embryonic notochord, and presumably with a very specific epigenetic state and epigenetic dependencies. That's a very unique cell, and it is not supposed to be there in the adult. And we know from published work that modifying the histone marks by certain drugs like histone deacetylase inhibitors can induce apoptosis in chordoma cell lines. So somehow kicking that tumor out of its comfort zone in terms of the marks on the histones and the genes that are transcribed could induce apoptosis. Another thing, brachyuria is in a way an epigenetic controller. It travels throughout the nucleus and triggers transcriptions of genes. It's another kind of epigenetic phenomenon. And Dr. Hameed already described how brachyuria is sort of genetically implicated and is universally expressed in chordoma. And brachyuria knockdown induces senescence. So our question is, what is the state underlying epigenetic state, those histone marks, what does the code read like in chordoma? Does it vary a lot patient to patient, or is it all saying the one thing? Is it saying I'm a chordoma, or is it saying I'm almost a notochord cell? I mean, these are open questions. So we want to know what that signature is. Is it driven by mutation of specific chromatin regulating genes? Is it different from patient to patient? Is it held intact when you take a tumor out and make a xenograft or you make a cell line? And which cell lines match which patients in that space, the epigenetic space? And can we identify more specific targets for epigenomic therapy? So this is work actually on the website for the Chordoma Foundation from the Chordoma-Genomic Project, and I think you'll hear more about that later. I'm just absolutely so grateful to the foundation, but also to these researchers for early release of data because it helped drive things forward for us. And we were just looking at the few mutations that seem to hit these genes. We added to those mutations that came up in Dr. Hameed's impact panel, working also with Meera Gounder, and these are his patients' data. And we marked out all of the chromatin modeling genes that were hit by mutation, and we went to the literature and we identified the published hits as well and merged them together. This is Nelson's work. And we can map these out where the hits tend to be on a map of histone modification. We can start to make some predictions. So first, are these genes important at all? Well, they are important in other cancers. So here's just a table showing other cancers with alterations in these same genes that I boxed out in red, or I should say Nelson boxed out in red, and that they're implicated in other cancers. So when we look at the tally of mutations, none of them stand out. It's just like we see in primary GBM by tumor type, 2 out of 29 cases. The only one that's really prevalent is brachyury. That's the T gene. It's a funny name, but that's it. And we can make predictions about what those mutations might do at the histone level. So what I mean is, a chromatin remodeling gene is a gene that's designed, or the protein is designed to sort of travel around and identify certain histones in certain contexts, let's say important for being a notochord cell, and then to attach on little decorations, methyl groups, acetyl groups, or to take them off, and thereby to open up the chromatin and make it transcribe or to close it. And so if we know a little bit, we're learning more and more about what each of the enzymes does. So if we take that literature, we can start to make predictions. Okay, if this patient has this one mutant, maybe we can tell by looking at their DNA, and we can see that gene is not decorating the histones correctly. That would tell us maybe it's an important mutation. So we want to take these predictions to chordoma tumors. That is, if they have a certain mutation, that we're going to see modifications in one of these histone mark sites. And just to give you a key to read this, H3K27 means the histone 3 family. That's one of the subunits of the histone, and the 27th amino acid. All right, so that's how we read these. And at the same time that we answer that question, we also want to know, we want to just take a moment to take a broad view so we can pick up any signatures. We don't want to look at just one histone, one site. We want to get the whole snapshot if possible, because that whole snapshot may distinguish one set of patients from another set of patients. One set of patients who can respond to a histone demethylating drug and another that doesn't. So this is work in progress, and what we did is reached out to colleagues who found a way to take these histone sequences, and each of these boxes here marks one of the sites, like K4, K27, that gets decorated, and these colors are the decorations. Could be methylation, acetylation, et cetera. So how do we chop up the tumor and then measure each of those decorations? So one way to do it is to chop up the protein itself, take out the histones, chop up the protein into lots of little peptides, and then weigh them. And if you do that with a mass spectrometer, the ones that have a lot of decoration will be heavy. And the decorations are methyl groups, acetyl groups. You know their weight. So depending on their weight, you can tell how many methyl groups are attached to that histone at that location. At least that's what they tell me, because I have no idea how to do it, but we got Neil Kelleher and his group at Northwestern to do this with tumors. And we were very excited. This was, at least was verbally reported to us, the first actual tumors that they did this on, first tumors, solid tumors of any type. They'd been working mostly with cell lines. So we asked them to look at chordomas, and then to also, they ran some control cell lines. So the chordomas here are in red, the control cell lines are in blue, and this is a K27 site. So on the H3 histone piece, the 27th amino acid, does it have no methyl groups, one methyl group, two methyl groups, or three methyl groups? How decorated is it? And what you can see is that the chordomas here in red have more of the two methyls and three methyls. They got more methylation. And conversely, on the 36th amino acid, they have less methylation. And we know from others' work that that pattern matches repression. So these have a very specific program of suppressing gene expression at both those marks. So that would make sense, because it's not an undifferentiated cell. This is a cell that looks like a chordoma. We broke them out, because this is the exact same data, but now you're looking at each of the tumors individually, and I won't spend too much time because we're still doing the correlations here, but we have one clival case, we have the rest are sacral, and one dedifferentiated. And we have genotyping on one, but we've submitted these all for sequencing that's come back, and so we'll have genotyping on the rest. But overall, we can see that a pattern is actually very uniform, as if the majority of these have the exact same methylation and modification pattern. And we would make a prediction in this one patient who had mutation of KDM2B, which is a histone remodeler. We would have predicted more methylation at this K36 mark and more permissive gene expression in that patient as a result of that. And sure enough, we did identify higher levels of 3 methyls and very low levels of 0 methyls. So most of the histones are highly methylated. The H3K4 is an extremely permissive site. It marks where genes are being transcribed actively. And we didn't know what to make of this data at first. We expected H3K4 to be elevated because brachyury is activated by H3K4 methylation and also induces H3K4 methylation, at least in the literature. And so when brachyury turns on, it turns on many other genes. When we compare this with cell lines, the picture is quite clear. H3K4 is more active in these chordomas than it is in the pool of cell lines. Now remember, these are professional cell lines. They grow really well in the dish. And these are chordoma tumors taken out of patients where they were often or almost exclusively indolently growing. So that's really remarkable. And what we want to do is look at the genes that are specifically turned on at those H3K4 sites and see whether they can be modified by modifying brachyury expression. So our ongoing research, we're filling in the genotyping gaps right now to identify which chromatin modifying genes are mutant and which patients' tumors. We're doing exome genotyping to widen the net cast as well, and extending to DNA methylation sequencing, where we can start to see the signatures of H3K4 methylation. And performing an shRNA screen, it's a completely different approach to try to identify which of the chromatin modifying genes the tumor may be most dependent on. And in summary, chemical compounds, undeveloped drugs, and developed drugs exist that alter the epigenome in specific ways. There are several out and FDA approved now for other cancers, and there are dozens in clinical trial. There are compounds that affect histone methylation overall, DNA methylation overall, specific H3K4 methylation, and brachyury expression or its effect. And we need to understand the epigenomic landscape of chordoma to identify which of these approaches are promising and less toxic, and which models and cell lines match which patients. And we need to advise the drug companies which drugs need developing. So I want to thank my collaborators, obviously Nelson, but also Dr. Hameed, who provided us with the DNA and the annotation for the samples, and Dr. Gounder for his impact data, and Neil Kelleher at Northwestern. Thank you so much. Thank you.
I want to thank Dr. Brennan and Dr. Hameed. So we have next scheduled a Q&A with the two speakers that you just heard from. I know there's probably a lot of questions. We're running a bit behind, so we need to recover a little bit of time. And what I'm going to propose is that we just spend about 15 minutes for Q&A. So we'll cut our Q&A about 5 minutes short, and then we'll cut the break about 5 minutes short. And we'll aim to be back here at 11:10. So we'll just be about 10 minutes behind, and we'll try to speak quickly thereafter and recover a little bit more time. So if I could just invite Dr. Brennan and Dr. Hameed to answer any questions that you might have about their research. And Dr. Bilski will join us in the afternoon for the clinical Q&A. So we're going to have several other clinicians answer questions about treatment, about diagnosis, things of that nature. So for these two speakers, we're going to try to limit the questions to research-related questions. And there are microphones at the sides of the room. So if you have a question, please feel free to stand up, ask a question at the side of the room. And we also have people who will be running mics. So if you want to raise your hand, we can bring a mic to you. But please make sure that if you ask a question that you're on mic so that it gets recorded.
Is it possible for us to show pull up old slides from the previous last presentation?
I've got Dr. Brennan's slide. You mind if I go back?
Okay. About 12 slides from the end.
Can we get the slides up? There we go.
The one, it's going to be hard. You had one that had some red boxes.
Yeah.
So lots of us in the room have had lots of different kinds of things done to us, and we have family histories of lots of different cancers. And one thing that I saw when this flew by was AML seems to have things in common with one of those red boxes. And is that leukemia, acute myeloid leukemia?
It is, yeah.
So there's drugs for that were tried on lots of us, and it was successful for a while and had real impacts. And I'm wondering, can you correlate patient experiences with this kind of diagram, or is that way too premature?
Well, I guess we can, but until you know where to look to genotype. So that I would call that genotyping, and in a way, that's what the impact study does. That kind of panel where you can very efficiently take each patient and make these calls. And you can't do it across the whole genome yet. It's too complicated and expensive. But for instance, the impact panel here, every time one of these studies comes out, they add the new gene to the panel, and then it's tested in everybody. So I think maybe Dr. Gounder can comment on it later today. I think it's something that's being done very actively now on patients where we have the genotyping information. But it's always a problem of not knowing where to look outside of the lamppost. And I think not only are the gene mutations important, but for certain tumor types, and the leukemias are among those, it's not just the genes, it's the influence they have on the epigenome. And that's in the leukemias where epigenetic therapy is also being very actively investigated.
One other thing I just wanted to mention, so if you're looking at your agenda, you might have seen Dr. Gounder on the agenda. He unfortunately was not able to be here this morning, but is going to be here this afternoon, and he's going to be speaking this afternoon both about the treatment of chordomas using systemic therapy, so drugs, chemotherapies, things like that. And he's also going to mention some of the research that he's doing during his talk then. So just in case anyone was wondering, we haven't forgotten about Dr. Gounder. He's coming in the afternoon. Yes?
Chairman Willis. Hello. Is it on? Okay. I have a question about brachyury testing. And on the markers, my test was 5 years ago. Can it rearrange itself where your brachyury testing results would change, or is it always the same?
what we know about brachyury is its expression in chordoma. So it's a protein expression, so we generally see it consistently expressed in chordoma. We don't, from at least the studies have not shown real rearrangement of brachyury itself, but what it does are how other genes and growth factors attach themselves to the brachyury is variable, or it has some impact on how the tumor behaves, or maybe even how it forms. So generally, in chordomas, we see consistent expression of brachyury. Yes, we do see that.
So if you had an incidental clobal chordoma and your brachy was 0 and it hasn't changed in 12 years, do you get another blood test to see if your brachyury markers have changed? That would be a marker that it's growing? That's what I'm curious about.
Okay, so the way we actually look for brachyury is on the tumor itself. And we really don't know in the blood how it is expressed. It's possible the tumor cells can be circulating in the blood, and someday somebody would find a way to use it as a marker. That is all future research which will happen. But the expression of brachyury itself is within the tumor itself. So
the blood type test for brachyury is not necessarily an indicator.
No,
okay.
we don't. Yeah,
Thank you
it's not
very much.
indicative. Yeah, and as it is true that we expect brachyury to be expressed in all tumors, but nothing is 100% when it comes to cancer. Some tumors will defy everything that we know and will not express, and that does happen. But majority of chordomas express brachyury, and it's in the tumor.
Okay, thank you very much.
Just a comment. Given how rare our disease is, I'm just wondering how can we assure that samples be gotten from surgical sites for the gene sequencing that needs to happen in order to make progressions with all of the studies?
Well,
I just want to say
go
one
ahead,
thing
yeah.
to give credit to the pathologists and to the group here with Mike Berger and Mark Ladani, who developed this impact assay. They designed it from the beginning to work in paraffin. I'm a surgeon, and getting the fresh tumor is really an extra burden. And I don't mind the extra burden, but it involves more people and things can get lost, and it has to sit in someone's freezer. The safe thing is to get it out of paraffin, and that way any patient who comes can get the same genetic testing. I just wanted to thank you guys for doing that.
Yes, so now technology is advancing so much that we are able to use many different type of tumor samples. So in surgery, you get fresh tumor, which pathology receives, and we make sure that there is some part of it do get frozen away, for we don't know what future brings and how this can help in that particular tumor type. At the same time, for diagnostic purposes, the tumor will goes through a processing in the pathology lab. So I showed you those slides where you saw how the tumor looked like. We need that processing part. And now can we use, and that is like those samples are there forever. So 30 years from now, you still will have those samples. So when we advance knowledge in tumors, now you can go back and say, well, now we have so many samples there, let's study them, and that's how any advance in science happens. So as Dr. Brennan mentioned, you have those formalin fixed tumors, which can be used in these modern technologies, looking for gene sequences, looking for large amount of genes and mutations and other changes, which are now possible by what has advanced in the gene sequencing part of it. So we try to do it in every possible way. We take the tumor, if we can freeze it, we would keep some. We would use what is already available after the diagnosis for all the sequencing part of it. So, and the communication is between us and the surgeons. When Dr. Boland is operating on a chordoma case, he tells me, I'm doing it tomorrow. So I know about it, I alert everyone, when he sends the tumor down, call me. And so I know what should be done at every step. So that's how we are now preserving these rare tumors, because they are very precious. Sure, sure. Okay. Yes.
Thank you very much for both your lectures. They're excellent. In regards to specific DNA mutations which people with chordoma are found to have, is it recommended to have so I have 3 little boys, is it recommended that I do the genetic evaluation for them to see if they have the same mutations, even though I know that it doesn't guarantee an expression of the chordoma later in life? So what's the recommendations currently for offspring?
So,
Sorry, I'll say it louder. So basically, should we get our children tested for the genetic defects that are associated with chordoma?
well, that's so the SNP, which is found in association with chordomas, obviously gives a clue, perhaps, that there could be familial. We know about familial association, and those when it comes to children and you want to know something about it, I would say the first thing anyone should do is actually really talk to a genetic counselor about it. Every disease is different. What we know of associations and incidents and whether it's needed at what stage of a child's life needs a group of professionals who need to talk to the patient and family. And that's what I would say. It's a case-by-case basis that you need to discuss with the counselors.
And I don't know if anybody knows how practical it is to get that. I know specific genetic defects are easily found, but I don't know how practical that T mutation is. I don't know if you know, Josh, or...
I think it is really he was asking how practical this is to do this kind of genetic analysis. I would think it is really very much institution-based, and where someone has all the teams available to give that advice from genetic counseling to the physician who's highly involved in this tumor. That is the best way to go about any of these rare conditions and susceptibility gene testing.
I might just be able to add a little bit of extra color there. So just for everyone's clarity, the mutations that you heard talked about, with the exception of that T, or the brachyuria mutation, those are all mutations that occur in the tumor and therefore would not be passed on to one's offspring. The only mutation that is germline, or in other words inherited, is brachyuria. And there is actually a commercially available test for that. And some of you may have had that. Maybe there's actually a number of companies that offer it. And so if anyone is interested in learning about exactly how to have it done, feel free to speak with Shannon. We can point you to the exact location. I think the really important thing to keep in mind, though, is that SNP in brachyuria, that change in brachyuria, is really common. So 40% of the population has it, and yet only 1 in a million gets chordoma. So the likelihood, even if you've got the SNP in brachyuria, and probably everyone in this room does, every chordoma patient in this room does, the likelihood that your children would get chordoma is still, so the risk for the general population is 1 in a million. The risk, if you've got the SNP, is 5 in a million. So it's still extraordinarily low.
I had a question in observing some of the information on chordoma. Even though the aberration occurs when you're an infant and the cells break off, why is it that it takes up to the 6th decade for it to show up? Is there a correlation between hormones? That was an observation. For instance, for males, testosterone goes down, females, estrogen goes down. Is there some correlation between that? Because all of a sudden it seems like more people, then it starts manifesting itself when they're 50s and 60s.
Well, All I can do is comment on how, for instance, in glioblastoma, the other tumor type I showed, depending on how the tumor started, it could kind of start quickly and grow quickly and end up as a pediatric presentation, sometimes young adult, sometimes older adult, and that those, it wasn't just chance, it was that the particular way that the tumor grew meant that for the ones that show up in the older adults, they take longer because they take longer. That's all we can say. Does that make sense? That something about the way that the tumor develops takes longer. Now, on the route to becoming an older adult, you pass through puberty and you have passed through menopause, and so I think it's very difficult to tease apart the independent effects as a general statement. But Dr. Hameed?
When we talk about notochordal rests, that is, it's a structure which really we don't need to have. It should have disappeared, and nobody should be developing chordoma, period. That's how it should have been. But just like vestigial organs, we have appendix, which we don't need, we have. So there are cells which remain in the body from birth, and they have, the benign notochordal rest that actually shows as an abnormality in the radiograph do not always progress to chordoma. Just because you have a benign notochordal rest does not mean you will develop a chordoma. It's extremely rare that that will go on. There is no real proof that we know that notochordal rest means you will develop a chordoma. That we don't know, actually. We don't know why those things remain, and people can go through a normal life, and die of other natural causes, and have these sitting in their vertebra. Whereas chordoma, when it occurs in an individual for many genetic reasons, perhaps, undergoes certain mutations or certain changes in the genome and develops that malignant tumor. So it is to think that there is, why do we develop it after a certain age group when we have this sitting in our vertebrae from childhood? We really don't know that these cells which are sitting there are the ones which are turning malignant. But we only know they look like each other. They have the same expression of like brachyury. So there is some relationship, but the connection between the benign notochordal cells and chordoma is still not well established. And perhaps in the future, research may show there is a connection or some genetic change which will tip the balance and lead to a tumor formation in a susceptible adult. So one should not think we have a notochordal rest here, you have a tendency to them. It is not so. We really, most of them do not.
So lots of really good questions. We've got time for one more, but I know that Dr. Hameed and Dr. Brennan, I'm sure would be glad to answer questions during the break. So we want to go to break after one more question, but if you have more questions, please feel free to stick around. We can continue the conversation. So maybe over there.
We're incorporating a question because it kind of relates. When you mentioned capturing fresh tissue, which you were just discussing, for those of us who have recurrences, which there are a lot, and we're living longer and longer, so there's more time in between. And when I was first diagnosed, none of this, there was no foundation, there was nothing. So if you harvest fresh tissue, first of all, you need a surgery, which is Dr. Bilski was talking about, is sometimes not happening anymore. The old tissue, is 10 years old and God knows how usable. Is there a difference between the original tumor sample or a new one? For example, would a lung tumor be the same as the clival was? And the incorporation from the other question, which is one we all have really, is we've heard that decalcifying tumors, when they're saved, makes them unusable for the tests, so that's kind of a big question too.
Thank you.
Who wants to take over?
So from old tissue to when tumors recur, it is believed they do acquire additional genetic changes, especially if they are going to the lungs, which means they have metastasized, and generally metastatic tumors from the primary tumor. The reason they have been able to travel from the original where they arose to another place tells us that that has acquired some other changes to be able to have that property to go from one place to another. So there will be changes. We may not know what they are, but there will be changes. Second, so it is, if it's possible, generally it's if somebody will get operated for a metastatic tumor, we would acquire that tissue and freeze some and keep for further studies because we want to understand the disease. Second, about decalcification, yes, that is if you decalcify any tissue, that is not very good tissue for any type of DNA or RNA.
When bone is involved, just so people understand, that's not every cancer tumor, but specifically chordoma because there's bone involved. So
yes.
it's an issue.
So chordomas occur in the vertebrae. So it's bone is a very hard tissue, contains lots of calcium. So when it comes to pathology, if you take a bone and put a knife on it, you cannot cut it because it's a hard rock. So we have to remove the calcium from the bone, and that process is called decalcification. We usually use some form of an acid to decalcify bone, but acid is also a chemical which will destroy DNA and RNA. So what we try to do when we receive tumors, wherever there is no bone there is tumors grow out of the bone, right? We know that. So those parts of the tumor which is not in the bone, we would take them out and not decalcify that, so that's available. It may not be always possible. Somebody may have a tumor which is completely confined within the bone, and then we may not have a choice but to decalcify. But we are looking into other means to decalcify. There are chelating agents which can take away the calcium without destroying the DNA, but that takes a longer time to do that. But those are areas that we look into and see how we can preserve at least to an amount that we can use for future studies.
A patient, maybe not here, needs to tell their doctors ahead of time to look into that so that it can be saved the right way for testing.
Right. And
thank you for very good answers.
Sure. Well, please join me in a round of applause for Dr. Brennan and Dr. Hameed.